Hello, everyone. I'm Kit Rowe, Vice President of Sales and Marketing at Tiger-Sul Products. Tiger-Sul Products is a global leader in sulfur bentonite. We've built a reputation on quality products, extraordinary customer service, along with our commitment to research and education. On behalf of the entire Tiger-Sul team, we thank you for joining us on this webinar. Sulphur, a diverse product for diverse crops. This webinar is structured a little differently than most traditional webinars. It is in a question and answer format, as our two presenters will ask a question and the other will answer. These questions are an accumulation of those we've received over the last few years. Our intent is to address those concerns that tend to be the most prevalent inquiries. Hopefully, their input will generate additional questions from you. So now, I'll introduce our two presenters for today's webinar. David Annis is a Market Development Manager and former Technical Agronomist with Tiger-Sul Products. He works with fellow agronomists, dealers, and farmers to help them manage their soil fertility and maximize their profitability. He conducts nutrient and crop management trainings and publishes newsletters and technical bulletins on applications of sulfur bentonite and micronutrients. David received his Bachelor of Science degree from the University of Arkansas in Plant Protection and Pest Management and his Master's of Science from Arkansas State University in Agriculture. His professional certifications include Certified Professional Agronomist and Certified Crop Advisor. David has focused on multidisciplinary, practical agronomic solutions, research and education for over 30 years. Prior experience includes Texas A&M University, Noble Research Institute, and University of Arkansas in the areas of agronomy, entomology, plant pathology, plant growth regulators, and precision agriculture. Wesley Haun is a senior agronomist with Tiger-Sul Products. He coordinates soil fertility and crop nutrition research with various research organizations in the United States, Canada, and internationally. He conducts agronomic educational programs that emphasize the benefits of a complete plant nutrition management program. Wesley received his Bachelor of Science degree with a major in agricultural mechanization and a master's degree with a major in plant and soil science from the University of Tennessee. His professional certifications include Certified Professional Agronomist, Certified Professional Soil Scientist, and Certified Crop Advisor. He has a diverse agronomy background that spans retail, sales, and consulting at the farm gate to agronomists at regional, national, and international levels. So, without further ado, I'll turn it over to Wesley and David.
Thanks, Kit. I am honored to be a part of this program today. Sulfur addition to plant nutrient programs has become an important consideration. My objective today is to share information on different forms of sulfur, plant uptake of sulfur, sulfur deficiencies, and applying sulfur as a soil amendment. Yes, sulfur is a diverse product, as there are multiple ways it can be utilized in crop production.
There are several uses for sulfur. We can enhance crop production through the use of sulfur as a plant nutrient, as well as a soil amendment, and as a plant fungicide. Now, our products are not labeled as a plant fungicide. Therefore, we will focus today on talking about sulfur as a plant nutrient and as a soil amendment. Sulfur's role within the plant is quite varied. It's involved in 2 amino acids, which here again adds to the proteins. They're involved in enzymes. It's involved in the photosynthetic process and is a critical part of nitrogen fixation in legume plants. There are multiple forms of sulfur. That includes the sulfates, the thiosulfates, the bisulfites, polysulfides, as well as elemental sulfur. So David, how would you explain the difference between these sulfur forms?
Well, Wes, sulfur, basically, when we're talking about it, we're looking at sulphate, thiosulfate, and elemental sulfur. So ammonium sulfate, everybody pretty much has got experience with this. It's a 210024. Basically, they create it with the ammonia and sulfuric acid. It's water soluble, and it really likes to pull water in from the atmosphere. So anytime the relative humidity is over about 80%, you'll see it get wet. Now, ammonium thiosulfate is a 120026. People normally call this either ATS or Tiger-Sul. It's a white crystalline powder, water soluble, and it's real interesting because it's sold distributed as a liquid product. Now, it's 1/2 sulfate and 1/2 elemental sulfur. Usually when we're talking about elemental sulfur, we're talking about 99.5% sulfur. And it can be mined from natural reserves, but now we're pretty much recovering it from petroleum and natural gas in the refining process. It's dry, it's crystalline, we can store it outside, it's not water soluble, but when we heat it up to around 250 degrees Fahrenheit, it becomes flowable. We call that molten sulfur. Wes, tell us a little bit about the difference between Tiger-Sul's elemental sulfur.
Be glad to. Tiger brings in sulfur in the molten form, and we mix that with bentonite clay. People ask, well, what's the purpose of clay? Well, bentonite clay has high water absorption properties. Bentonite clay is in each pastel. As we bring it in a molten form, it's flowable. We are able to mix that such we get the clay evenly distributed throughout the batch. And once this bentonite clay product is applied to the soil, it absorbs soil moisture, it swells, and then fractures the sulfur into small particles. The bentonite clay changes the dynamics of the sulfur product. Therefore, its functionality is significantly different from pure elemental sulfur. So in application management with sulfur, spring applied application is recommended for sulfate products and the ammonium thiosulfate products. With sulfur bentonite products, we can apply that in the fall or spring. So what is the parts per million of sulfur do you like to see in a soil test, David?
Wes, as a general rule, the short answer is anything less than 15 parts per million, I say that you have a sulfur deficiency. Now, the soil test value is kind of like a snapshot in time. It's going to tell you what you've got out there. You've got to remember that sulfate, like nitrate, can move with the soil water into the soil profile. Now, the soil sulfate value is constantly changing, and this is due to the microbial activity. We prefer to see plant tissue analysis rather than soil sample analysis. The plant tissue analysis, we feel, gives you, it's the gold standard, gets you a much more complete idea of what's going on in the plant at that time. We took a look in 2020, we took a look in the Mississippi Delta out of 52 soil samples, and these were on some very well-managed fields. We found that 81% of them were below 10 parts per million. Now, if you remember, I told you that anything less than 15 parts per million, we feel that you need sulfur out there. We only had 13% of those, but 81% were below 10 parts per million. And this was kind of a shocker to us. Well, Wes, when we talk about sulfur bentonite oxidation, how fast does Tiger-Sul sulfur break down in the soil? And will it be available in the first year?
Well, that is a very popular question. And in order to answer that, I like to review the sulfur cycle briefly. As we look at this in the upper left-hand corner, we see the plant. And here again, in the fall of the year, plants mature. At harvest, the residue goes back to the soil. Bacteria breaks down the residue, and we have soil organic sulfur. And it's acted on by bacteria and converted to hydrogen sulfide gas, which is at the top of the graph. And if this is on the surface, we lose that gas into the atmosphere. If it's in the soil, it's trapped and acted on by bacteria again and can be converted to iron sulfide, manganese sulfide as an example. Or it can be converted directly to elemental sulfur. And then the elemental sulfur then is acted on by a group of bacteria, and thiobacillus is the primary one, and converted to sulphate. Sulphate's at the bottom of these schematics, and notice sulphate is the form that the plants can take up. And any excess that may exist, and we have excessive water, we can see that sulphate leach down the soil profile and out of the rooting zone for the crop. The top part of this schematic is actually the mineralization process, and lower right-hand section of this is actually the oxidation process. Now, to look at some of the chemistry, and very briefly, starting with hydrogen sulfide gas is H2S plus your oxygen with bacteria, we obtain water and elemental sulfur. Take the elemental sulfur on the second line, in the presence of oxygen, water, and bacteria, we get sulfuric acid. That sulfuric acid then will react with the calcium carbonate that's in the soil and produce calcium sulfate, as well as carbonic acid. Carbonic acid is a weak acid. It will dissociate into water and carbon dioxide. Now, the important part of this whole reaction is the fact that sulfuric acid is produced. If there's enough of the sulfur, then we can potentially lower the soil pH. Now, for all this to take place, we must have these 5 factors involved. Soil temperature. The optimum temperature for bacteria to convert elemental sulfur to sulfate is about 65 degrees to about 95 degrees Fahrenheit. That's approximately 18 degrees up to 35 degrees on the Celsius scale. Soil moisture, that's going to be slightly below field capacity. So we must have optimum moisture there for the bacteria to function. Since the primary bacteria that converts elemental sulfur to sulfate requires oxygen, so we must have good aeration within the rooting zone, and then the microbial activity is a function of the population. The higher the population, the faster this reaction takes place. Last, but certainly not least, is a sulfur particle size. The smaller we can get that sulfur particle, the faster the microbes can convert elemental sulfur to sulfate.
And how does that influence sulfate release?
I'm glad you asked, David. I think there's a very distinct difference we need to emphasize here between elemental sulfur, which is pure or almost pure, 99.5%, whereas sulfur bentonite contains 90 or 85% sulfur. And in those products that we add micronutrients, that sulfur concentration will be somewhat lower. The distinct difference is in the name bentonite. As we add bentonite clay to the product, we want to enhance the functionality. Notice on the left is Tiger-90 CR. It's in a pastel, very consistent size. On the right, we have pure elemental sulfur. This is in a wide range of particle sizes. And notice, by looking at the ruler there, we've got some particles that are at least 1/4 of an inch in diameter. Here again, with elemental sulfur, it's not water soluble. It does not break down in the soil very fast, except for the very small particle sizes that you see there. The larger ones will take several years to actually break down. So by adding the clay, we fractured this Tiger-90 product into much smaller particle sizes. And we went to the lab to actually analyze some of these particle sizes. After that pastel fractures and breaks apart, we wanted to take a look at just to what extent do we obtain varying particle sizes. And notice on the left, we have 210 micron sizes. And you look at those individual particle sizes, you can see they are of size that still can be broken down by the bacteria. The 149 micron size is the size that we tend to anticipate being able to oxidize and convert from elemental sulfur to sulfate in the year of application. This next slide has the 149 compared to 105 microns. And you notice they're getting even smaller sizes here. The 105, we expect to have a high amount of oxidation in the first year of application. And to help put the 105 micron size in perspective so that you can help visualize that a little better is 105 microns, about the same thickness as a sheet of notebook paper. So we want to take this one step further, is to go to the lab and actually analyze these different particle size groups to assess the release rate at which these particles can be converted to sulphate. But before we can do that, we wanted to look at just the particle size comparison. If you take one cube that's 1 foot by 1 foot by 1 foot in 3 dimensions, you got surface area of 6 square feet. It's one cubic foot. Okay, if we take one surface area of the 1 cubic foot and then divide that 4 ways, so you'd have a half a foot width and height. And notice when we do that, we get 8 particles instead of 1. And we double the surface area from 6 square feet to 12 square feet. Okay, with the surface area at 12 square feet, we take one of those cubes and we divide that 4 ways. So when we do that, we increase the particle size, or increase the number of particles to 64. And then we double the surface area again to 24 square feet. So here, this serves the same principles we're trying to accomplish by adding the bentonite clay into our product, is to increase the surface area so that the bacteria can feed upon the sulfur at a much faster rate and convert it to a sulphate. Going back to the lab and the 3 different particle size groups, the 210 microns, the 149 microns, and the 105 microns, with 2 of our products, the Tiger XP and the Tiger 90. If you notice, the release rate is average about 80% of each amount of each particle size group. And this release rate was for the first 6 weeks of the trial. Now, we did 3 laboratory trials. This is the average of all 3 trials that were conducted. And here again, very consistent between the products. So what has led to sulfur deficiencies the past few years?
Wes, that's an excellent question. This diagram, or this chart, shows sulfur deposition in 1986. Now, this is in kilograms per hectare. And if you notice, we're looking at about 24 pounds or about 20 pounds per acre of sulphate being applied. Now, this is free sulfur that the farmers and ranchers were getting. And if you notice, most of it was on the eastern half of the United States. In 1990, the Clean Air Act came along by the EPA. And what it did is it started regulating sulfur deposition. Even now, we take a look at the diesel and we're looking at low sulfur diesel. Now, as we look in 2019 in this slide, this shows the sulfate ion wet deposition across the United States. If you notice, all these numbers in here are less than about 10. Some of them down to 2. But this gives you an idea of how much that we have reduced the amount of sulfur being deposited on our land. Now, what does this mean for us? Well, we're seeing more sulfur deficiency trends. This is something that we never were really taught in college. Sulfur deficiency was not going to be a problem. It was going to be acid rain. But with intensive management, high yielding crops, erosion, and the addition of no more sulfur fertilizer, what we're starting to see is sulfur deficiency. We've mined the soil from sulfur. Here's another question I've got for you, Wes. Crop uptake versus crop removal of nutrients.
Yes, David, and that is a very controversial question oftentimes, but we like to look at this from the standpoint of what that crop actually needs. So from an uptake standpoint, it's the amount of nutrients that a crop requires to grow and maximize its yield potential. Crop removal is the amount of nutrients that leaves that field at the time of harvest. So uptake. crops' uptake rates are higher than crop removal rates. Those removal rates can cause some yield drag, and sulphate reacts similar to nitrate in the soil environment. So we need to supply sulfur in sufficient quantities to meet specific crop demands. So I have in this chart a few of the crops that we work with on a daily basis. And you have the crop, we have pounds of sulphate per unit, yields per acre, then the uptake rate of sulphate versus the removal rate. So let's take a look at two of these crops. The first one being corn. The uptake is 32 pounds of sulphate to obtain 175 bushels of corn. But when we harvest that corn, there's only about 14 pounds of sulphate removed from the field. For example, we look at cotton. 15 pounds of sulfur required for each bale of harvest. And if you're expecting 3 bales per acre, that uptake requirement is 45 pounds of sulfate. And the removal is only 15 pounds. Going back to the corn, at 32 pounds of sulphate per acre required to raise 175 bushel, that equates to 36 pounds of Tiger-Sul that needs to be applied to accomplish the 32 pounds of sulphate. Whereas with the cotton, with the 45 pounds per acre of sulphate required, that would equate to 50 pounds per acre of Tiger-Sul to give you the equivalent amount of sulfur that's required. We need to keep in mind that using the uptake rates instead of removal, if we make a recommendation based upon removal of corn, for example, at 14 pounds, there's a good possibility to have a deficit for that corn crop. And the same will be true with all the other crops as well. Something we need to keep in mind when we think about this is, here again, sulfate reacts in the soil similar to nitrate. When we think about phosphorus and potassium, they become part of the cation exchange complex. And we can make recommendations based upon removal for those two nutrients. But since the sulfate is mobile within the soil and it is more difficult to predict the carryover potential, there's no, here again, interaction in the soil from the standpoint of a part of the cation exchange complex. So we need to look at this from the standpoint of what that crop needs rather than the removal aspect. Uptake of sulfate and all the essential elements is a function of the availability of the nutrients and the proximity to the root surface. We strive to maximize crop yields. Oftentimes, what is out of sight is out of mind. We tend to overlook the dynamic soil interactions. Those include the physical, the chemical, and the biological. These interactions influence the availability of nutrients for uptake by crops. The first point of nutrient uptake is the interaction at that root-soil interface. Nutrient availability at that root-soil interface is controlled by the soil properties, root characteristics, crop growth, nutrient interactions in the soil, and soil microorganisms. All these interact at that root-soil interface referred to as the rhizosphere. This photo shows the roots and the little root hairs projecting laterally from each root, and it's that zone within where the root hairs are located and the root itself is actually called the rhizosphere. A number of management factors that farmers have direct influence on are the plant density, soil nutrient availability, the planting date, and IPM, and there's probably other factors you can add to this list. So David, how does a winter with a lot of freezing and thaw or wetting drying cycles affect the conversion of sulfur bentonite to sulphate in the spring?
When we talk about freeze thaw, wetting and drying, it's an indirect relationship. Basically, what we're trying to do is fracture the Tiger-Sul product into smaller particles. These smaller particles increase the surface area and provide more area for the soil microbes to feed on the particles, converting them over to plant-available sulphate. The larger number of particles, the greater the feeding area for the bacteria. The more feeding area, the larger number of bacteria, resulting in faster sulfur to sulphate conversion. Wes, what are some of the sulfur deficiency symptoms we may observe in some crops?
Sure, I'd be glad to describe some of these deficiency symptoms, and I have some photographs here to share with you. And we'll start with corn. We got to keep in mind that sulfur is mobile in the soil. It's immobile within the plant. So with those nutrients that are immobile in the plant, the deficiency symptom shows up initially in the new growth part of the plant. So in corn, we're talking about the whorl, and we see some general yellowing of the plant in the whorl, and sometimes we see some striping take place as well. In cotton, here again, it's going to be in the new growth leaves. We have a general yellowing effect on those leaves. This was a soybeans. On the left, you see a healthy plants compared to sulfur deficiency on the right. And again, it's in the new growth is where we predominantly see the yellowing effect in the leaves. Potatoes gives us the classic symptomology of sulfur deficiency. Notice how bright the yellow is in the new growth leaf tissue, and the lower leaves still have their dark green appearance. In wheat, on the left, we have a close-up of the wheat plant. Notice it's in the top part of the leaf or the plant that we have the yellow discoloration. And on the right is a overall view of a field showing the areas of sulfur deficiency. And that's something else we need to keep in mind when we observe a potential sulfur deficiency symptom is it's not the whole field. It's going to be in various areas of the field. And it's not unusual to see it mostly on side slopes and hill points within a field due to either erosion type effects or high productivity.
Can there be any yield reductions when sulfur deficiency symptoms are not present?
Wes, that's a good question. Hidden hunger is the main thing that we look for in this. Now, hidden hunger is where the plant and crop looks normal. We don't typically see any deficiency symptoms out there. However, where it comes back to catch you is on the yield production or the growth of the plant. It's going to be less than what's expected. This is the chart that we learned about in college when we were discussing hidden hunger. On the left side of the chart, we're basically in deficiency. We see visible symptoms. We are affecting our yield negatively, and then we move into an area that's not quite sufficient, but it's not really deficient either. We're not seeing the symptoms, but we're still negatively affecting yield. What we want to do is we want to meet sufficiency of plant nutrition. And that's what we're shooting for anytime that we're out here making fertilizer applications. This is a cotton field in 2020. As you can see, I've lined the rows up between a treated and non-treated. We see the treated, the cotton looks healthy. And we look at the row spacing in the deficient cotton, and we can see middles haven't lapped. We're looking at this, and we can see also a lighter colored yellow cast to the top of the crop. So this is showing the difference between a treated plot of cotton and a deficient or untreated. Now again, this is in 2019, and you can see just almost to the row where the treated and untreated is. This is 2 days after defoliation, and you can see that the untreated check, we've dropped a lot of the leaves already off of it. However, on the Tiger 90 treated, we see that the plant is a lot more vegetative and a lot more bolls on it. Wes, is there an advantage to a specific Tiger-Sul sulfur placement?
Yes, David, it's interesting that you asked that question. I do have a set of data where we've evaluated applying Tiger Zinc 18%, Tiger 90, and an untreated control in 4 different application methods. That is a 2x2, which means 2 inches to the side and 2 inches deep beside the row at planting, in the furrow with the seed, and then side dress and as a broadcast. We conducted this trial for 3 years at one location. And the values you see in this chart is an average of those 3 years of data. I might direct you to the 2x2 application method. It offers the most consistent response between both the Tiger Zinc and the Tiger 90 at 193 and 195 bushels per acre compared to the untreated control at 182 bushels. The in-furrow application shows 183 to 186 compared to 183 on the untreated control. Now with the side dress application method, we see a slight increase in both the Tiger Zinc as well as the Tiger 90 in comparison to the control at 189 bushel for the Tiger 90 and 186 bushel per acre for the Tiger Zinc, compared to the untreated control at 182 bushels. So that's about a 6 bushel difference. And then in the broadcast application, this is of course applied prior to planting, and the Tiger Zinc gave us a significantly greater yield response at 198 bushels compared to 182 by the untreated control, and Tiger 90 came in at basically 10 bushel less than the Tiger Zinc at 189 bushel. So hopefully this provides some insight as to the different application methods. We are confident that either method works depending upon each individual farmer's management and equipment he has to work with.
Wes, in a legume crop such as soybeans, what's the best diagnostic method to determine if sulfur deficiency is resulting in reduced nitrogen fixation?
Symbiotic nitrogen fixation is a process where rhizobia bacteria infect the roots of legume plants. They form structures we call nodules. It's within those nodules that the rhizobia converts atmospheric nitrogen to a ammonium which the plants can utilize. And hence, reason we call this symbiotic is the fact that the nodules and the rhizobia supply nitrogen to the plant, whereas the plant then supplies carbohydrates for the bacteria to live. So when the activity within those nodules is less than desired, we have a deficient sulfur situation that results in less nodule formation. There's inhibition of nitrogen fixation and a reduction of the nodule metabolism. Here we have some photos of nodules on a soybean root. Notice on the left there's a number of nodules. If you look closer and slice open one of those nodules, and if there's a bright pinkish red coloration, that's an indication that the rhizobia are active in converting nitrogen. You notice there's some there that's a whitish pink, so those are inactive, and bacteria are just forming in those to start the nitrogen conversion process. Now, if that area of the nodule is a brownish color, that's an indication that the bacteria have deceased, and actually those nodules only last about 30 days, so they will slough off and new nodules will form and continue the fixation process. So David, this year has been a year of extreme weather conditions. We've seen drought conditions and we've seen extreme amount of flooding take place. Can you expound on some of the extreme weather conditions and how that may influence the sulfur interactions in the soil?
Yes, Wes. What happens to the sulfur bentonite in a drought type of situation is that we're not seeing a lot of conversion, and that's due to a lack of soil moisture, a lack of microbial activity, And again, that leads to less sulfur oxidation and less conversion from the sulfur bentonite over to the sulphate that's plant available. Kind of as a rule, if the plant is actively growing, then probably the microbial activity will be actively converting. One of the good things about this is because there's less sulphate being formed, then if we do get rainfall in the wintertime, we have less of a problem with leaching. And we have less sulphate uptake by the crop. Now, this leads to a greater potential for carryover of sulfur bentonite to the next year or to the next crop. Sulfur and drought. Sulphate moves in the soil with moisture, and basically just like nitrate. A lack of moisture means less sulphate uptake. Will the sulphate remain in the soil the next spring? That's going to be a function of how much moisture or how much rainfall we get during the winter. But the sulfur bentonite, it's not going to be oxidized during the wintertime and should remain right where it is in the soil. In legume crops, less soil moisture is going to mean less nitrogen fixation. Again, less microbial activity. It's going to lead to poor nodulation in the plants, and it's going to lead to less nitrogen being formed. We're going to have some type of sulfur to stimulate the rhizobium activity. The thing I'd like to point out is sulfur increases the nitrogen use efficiency in the plant. Management for next year, since we have potentially less sulfur being utilized by the crops, we should see a greater sulfur carryover. And we may be able to cut back on our sulfur application next year. Thing we need to do is we need to take soil sample analysis in the spring to determine the sulfur application rates. Moving to extreme weather conditions like flooded soils, what happens in that? Well, when we're anaerobic, basically in a flooded type situation, what happens is we are looking at a different set of microbes to convert the sulfur to sulphate. When we're looking at sulfur reactions in flooded conditions, we're looking at a different set of microbes, anaerobic microbes, that will convert the sulfur bentonite to plant available sulphate. We do have a sulfur loss potential there. We have immobilization by microorganisms. We have reductions of sulfides. We could have volatilization, and we could have leaching. So our management for next year is we're going to have a limited carryover of sulfur. So what we need to be doing is applying the full uptake rate of sulfur in the following spring. And we're going to base this on the uptake requirement for that crop. Also, never hurts to take a soil sample analysis in the spring. Just remember, soil sampling is not the best method of tracking sulfur in your soils. It's more like a yardstick. Wes, how is the addition of micronutrients into Tiger-Sul products different from other micronutrients?
When talking about micronutrients, I think it's necessary that we relate to the soil pH and the availability of microbes. And in this chart, which I see you have probably seen numerous times, on the x-axis we have a partial segment of the pH scale. And as we look at this chart, you see the wider sections of each bar indicates a greater availability. But take note of all the micronutrients. All those tend to decrease as you go to a pH greater than 7.0, with one exception, and that being molybdenum. Keep this in mind that as the pH increases, we decrease the availability of micronutrients. So I wanted to bring this to your attention as we flow into the different types of micros. There are a number of different types of micronutrients. We got the chelates, there are sulfates, oxysulfate, and lignosulfonates. As far as the Tiger micronutrients, we utilize oxide products, and the question we often receive is, how does an oxide become available for plant uptake? For those of you who are familiar with micronutrients, realize that the quality of the micronutrients is based upon water solubility. And oxides are not water soluble. So just how effective is an oxide as it relates to Tiger products? We like to look at this from a microsite enhanced technology standpoint. We've got the larger yellowish orange colored particles that are representing the sulfur. Then the little yellow squiggly cues would be the thiobacillus bacteria. And then notice this little glow around the oxide particle. This is the reactive acid zone. This is the sulfuric acid that is created during the breakdown or conversion of elemental sulfur to sulphate. That sulfuric acid then acidifies that oxide particle and converts it to plant available form. Whether if it's a zinc oxide, you get zinc sulfate. If it's iron oxide, then we get iron sulfate. Those forms are then available for plants to take up. So as we look at Tiger products, that sulfur serves as a carrier for the micronutrient. We can add one or more micronutrients into the sulfur product. By doing this, we're able to capitalize on the sulfur oxidation process, which is a gradual release of the sulfate. And as the sulfate is converted, then we have a gradual release of the micronutrients. We get benefit from that standpoint as well as the distribution at the time of application. So David, do you have any field research to document the effectiveness of Tiger-Sul sulfur bentonite products?
Wes, yes, we do. I'd like to point out this is a corn response to Tiger Sulphur. Basically, we're looking at a yield advantage in this case of about 5.2 bushels per acre. Now, this is an average of 4 years, 4 states, and 74 trials. So it's not just a one-off. This is something that we're seeing numerical differences, positive numerical differences every year. As far as our wheat response to Tiger Sulphur, this is an average of 18 trials over 8 years. Between the Tiger 90 CR and the control, we're seeing a 3 bushel yield average. As we look at rice response to Tiger Sulphur, again, this is our Tiger 90. We're seeing that a yield advantage of 10 bushels per acre. Now, this is 4 years of data and 14 trials. As you can see, 10 bushels a year, current price of commodities, not bad. Now, as far as the tomato response to sulfur, we're looking at our untreated check, a grower standard, and the grower standard plus Tiger 90. This is 3 years, 1 location in California, but we're looking at a yield advantage of 3.8 tons per acre. As far as cotton, we're looking at Tiger 90, and we're also looking at Tiger Zinc. And one of the things that we're seeing, this is an average of 78 trials, 4 years in 4 states. Guys, we're looking at 129 pounds per acre yield increase. That's on the Tiger Zinc product. The fall application of Tiger products is highly encouraged because we get minimum sulfur oxidation in the fall. We can maximize our sulfur fractioning, increase the number of particles and the surface area, and we're optimizing the sulfur to sulfate conversion in the spring. Now, we're also increasing the plant uptake of sulphate in the spring, and we're maximizing our time use efficiency. So if we can apply it in the fall, that's one less thing I have to worry about during planting. Wes, could you tell us about sulfur as a soil amendment?
I sure can. And soil amendments are materials we add to the soil to improve its characteristics that are more conducive to root growth and enhance nutrient uptake. Some of the adverse conditions we have to contend with is an alkaline soil has a high soil pH. A textbook value is greater than 7.8. A saline soil has a high salt content. Sodic soil has a high sodium content. And then we can have soils that are a combination of the two with saline and sodic soil conditions. We have high salt as well as a high sodium content. So all these present challenges that are unique to their individual situations. In an alkaline soil with a high pH, you can lower that pH by adding an acid-producing product. What better product can we add other than Tiger 90? And the application rate of the sulfur depends on primarily the soil texture. Whereas a sandy soil would take less sulfur to make the conversion, whereas a fine textured soil which has a higher buffering capacity would take a higher level of sulfur application. Saline soils. There's no chemical amendments that we can add to change the situation. We need well-drained soils or soils with artificial drainage and an ample supply of water. It's going to take 6 to 12 inches of water in excess of what the crop will need to essentially flush the salt out of the rooting zone and move it down lower into the profile. Sodic soils may impact plant growth by specific toxicity to sodium-sensitive plants. We can have nutrient deficiencies or imbalances. We can have a high pH, and it'll cause dispersion of the soil particles that causes poor physical condition of the soil. Just which conditions are Tiger-Sulphur products effective? Those would be the alkaline soil, which strictly just a high pH where we can add the Tiger-90 and the reactions create the sulfuric acid and release the hydrogens and lower the pH. And the other would be the sodic soils. Almost always in a sodic soil, you have a high pH in conjunction with the high sodium. So by adding Tiger or a 90 product, we can lower the pH, and the oxidation process creates a calcium sulphate, which will dissociate. The calcium will displace the sodium, and then we can flush the sodium out of the rooting zone and move it lower into the soil profile. So David, what are some final thoughts you may have in reference to the questions that we've entertained today?
Wes, one of the things I'd like to say is, sulfur is an essential plant nutrient. It's like the fourth major nutrient, as it's been called. The bentonite clay, it's going to enhance the functionality of Tiger-Sulphur products. It's one of those things that you've got to have it to make it work. The crop sulfur uptake is significantly greater than crop removal, and we need to be fertilizing for sulfur uptake rather than crop removal. Hidden hunger of sulfur contributes to crop yield reductions. Can't say that enough. Yes, Tiger-Sulphur products enhance crop yield. We need to capitalize on the sulfur oxidation process. Put it out at the right time. We're looking at a gradual release of micronutrients into the soil. Over time, they'll become available to plants. Other than that, we'd like to thank you for being with us today, and for further information, contact your local Tiger-Sul account manager.
Thanks, David and Wesley. Now, a quick reminder, if you have a question, click on the Ask the Question button in the upper right-hand corner. Then type your question into the pop-up box and click the Send Question button. Here's a question from one of our webinar listeners. What were the Tiger-Sul sulfur application rates in the data?
That's a good question. I hate that I left that out. But on all the row crops, it was 30 pounds of actual sulfur on a per acre basis. The tomato one, that was 90 pounds of material, and that test was conducted in California.
Thanks, David. Here's another question. Wes, what is the significance of the nitrogen to sulfur ratio?
I use the nitrogen-sulfur ratio more from a diagnostic standpoint. As I collect a plant tissue analysis, I'm looking at the nitrogen to sulfur rate and what's in the plant. If I've got a ratio that is exceeding 15 to 1, I probably have got a sulfur deficiency, and that's going to be a function of the crop or species you're working with, because it can vary between various crops. For example, canola has a nitrogen sulfur ratio about 7 to 1, whereas in corn it's about 10, 12 to 1. So if it's greater than that, then you tend to have a sulfur deficiency. If it's less, then you obviously should have enough sulfur in that plant. This is not the rate at which you apply your nitrogen and sulfur product as far as the material application standpoint. Again, it's for a diagnostic standpoint primarily.
Do you recommend different application rates based on application methods?
Well, one of the things that we see is that either broadcast or banned, using the same rate is not really a problem. The reason for this comes back to the mobility of the sulphate in the soil. What we do see is if applied in a band, sometimes we may have a little bit longer before it is converted over to plant available sulphate. But all in all, either application is recommended.
Thanks, David. Here's another good one for Wes.
Is it safe to place Tiger 90 in the seed furrow or in the row?
Yes, Tiger 90, Tiger XP products can be applied in the seed furrow or row at the time of planting. We have a very low salt index. It's less than 4. With that, we don't affect the germination rate of the seed. And this, of course, is applying it at rates that are normally for plant nutrition in at 25 to 50 pounds per acre.
Thanks, Wes. Let's direct this question to David.
Is it acceptable to blend Tiger products with all other fertilizer products?
That's a good question. The answer is don't blend our products with anything containing nitrate. Nitrates are oxidizing agents and shouldn't be mixed with our products. Anything else, MAP, DAP, potash, I would encourage you to blend it with it. Save a trip across the field.
Thanks, David.
It looks like we've got time for one more. And I know this is a concern that I've heard personally when traveling with our sales team. In my area, we have trouble with low soil pH values. I'm reluctant to apply sulfur due to reducing the soil pH further.
I can appreciate your concerns, and normally with most fertilizer applications, especially with our sulfur products and the concerns of lowering the pH with the application rates. But generally, soils have a buffering capacity that will offset any acidity that you're going to create by applying our 20 to 50 pounds of our product per acre. And this is for plant nutrition needs. Now, if you want to change the pH or concerned about changing pH, it's when we apply a much higher rate of our Tiger product. So with that, I don't think you'll have any major issues unless you're applying it to a sandy soil with essentially no buffering capacity. Then you might lower the pH.
Thanks, Wes. In closing, I would like to thank all of you for joining our webinar today. A recorded version of this webinar will be available on our website. Anyone who watches and needs CCA CEU credit can self-report. If at any time you have additional questions, please visit our website. Once there, you can find your local account representative's contact information or simply click on the Contact Us link. If you include your name and email address, send us a message, we will respond promptly. You can also check out our website for additional information on sulfur bentonite products, as well as requesting access to Tiger University, where you can see white papers, technical bulletins, newsletters, and other valuable information. Thanks again for joining us today. Have a great rest of your week and an outstanding weekend.